Hello, everyone, and welcome to today's webinar titled Complete Toolbox for Gene Therapy Development. This webinar is part of the 6th annual event in the Bioprocessing Virtual Event Series. I am Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now let's get started. I now present today's speaker, Rumen Buguev, Head of Segment Marketing at GenScript USA. For a complete biography on our speakers, please visit the Presenter tab from the menu on the left of your screen. Rumen, you may now begin your presentation.
Thank you, Cassie, for the introduction. It is my pleasure today to present to you about the complete toolbox for gene therapy development from GenScript. We essentially offer a one-stop solution from discovery to clinical trials and clinical manufacturing. This is the agenda for today. First, I'll talk briefly about the gene therapy solutions overview of GenScript's tools, then our tools for target discovery and validation, our tools for gene therapy lead optimization and generation, and our tools for preclinical to clinical development and clinical manufacturing. So first, a little bit about the company. GenScript is a company that consists of several entities. For this presentation, I'll start from the right side. One of the entities inside GenScript Group is Bestzyme. That's our synthetic biology company. They focus on developing new tools for food technology, for animal feed and human food supply. The next is Legend Biotech. Legend Biotech is a cell therapy company. They recently launched their latest cell therapy, Carvykti, working with Janssen. They also have full pipeline of other cell therapies in development. My focus will be, however, on the other entities on the left side, which are our GenScript Life Science CRO division. That division also contains our Life Science Products division. Also our Biologic CDMO division, GenScript ProBio. So my focus today will be on the capabilities delivered by these two divisions. So GenScript has complete solutions for multiple applications. One of them is gene therapy. Another one is cell therapy. We have complete solution for antibody therapeutics, vaccines, and IVD, as well as some other applications such as synthetic biology and plant biology. Today, my focus will be on the gene therapy specifically. So every gene therapy starts with target discovery and validation. First, a lot of bioinformatics analysis is performed, a lot of patient samples are analyzed on gene level, protein level. After that, depending on the type of therapy, there are different types of therapy that could be developed. Some are ex vivo non-viral gene therapies, also sometimes referred to as cell therapies. Others are in vivo viral gene therapies, and others are in vivo non-viral gene therapies. In this presentation, I'll focus on the bottom two, which is our tools for in vivo viral gene therapy and in vivo non-viral gene therapy. So we have very broad portfolio with many solutions, products and services. Typical gene therapy and actually any therapy drug development starts with target discovery and validation, then proceeds to lead generation and optimization, then preclinical work, clinical trials, and clinical manufacturing. As you can see from this slide, we have complete portfolio of tools for every stage of the development. Target discovery, target validation, viral vector lead generation and optimization, non-viral vector lead generation and optimization, preclinical studies, IND, we provide IND support and also with IND filing. We also provide GMP manufacturing for clinical trials as well as drug manufacturing. So what are the tools for target discovery and validation? So in target discovery and validation, very typical process starts with gene level analysis. Gene screening, sequencing, very typical tools are being used like qPCR for up and down gene regulation analysis, next-gen sequencing for sequencing analysis, CRISPR gene editing for target knockout for validation. After the gene level analysis, very often is performed protein level analysis. For that, we have a lot of tools also for protein level analysis. We have very strong reagent antibody services for developing various antibodies for detection. We have protein expression services, sometimes for expressing targets and antigens. We have full portfolio of Western blotting products, and we have also very strong capabilities for cell-based assays. So the first basic materials are very often the qPCR related materials, primers and probes. And we have very strong capability in that regard. We can produce essentially any primer and probe. We can produce them under ISO 9001 or ISO 13485 quality systems if needed. We produce with very high quality to eliminate any non-specific peaks. On the right side here, you can see some results from various targets being analyzed, as well as positive and negative control. You can see that our primers and probe perform very specifically. We can also produce various gRNA libraries for gene knockouts. We have special platform called the Gene Titan platform. This is microchip-based oligosynthesis. This platform allows us to produce up to 8 million oligos in parallel, and we can produce them very efficiently with very low error rate. We have excellent coverage of 99.9%. We can produce them in different amounts. So from here, we can easily prepare gRNA libraries. Essentially, after we synthesize the oligos in parallel, we can PCR amplify them. We can clone them in the corresponding vector, and then we can deliver the gRNA libraries depending on your requirements. Could be delivered as a plasmid version or as a virus version. We also offer our libraries in various quantities from microgram level to milligram level, and we don't have any sequence restrictions of what kind of library we're able to make. Another tool is very much used for target discovery and validation are antibodies and used for various immunodetection techniques such as Western blotting, flow cytometry, IHC, ELISA, and so on. And we have very strong antibody capabilities. We can produce polyclonal antibodies, monoclonal antibodies, bispecific, single domain. We can produce scAVs, VHH fragments. We can also produce custom ELISA kits. We have multiple platforms for that, ending from traditional hybridoma to phage display libraries. Also, where we have excellent rabbit monoclonal platform. Rabbit antibodies are very well known for being very specific. So they're very often the antibody of choice for various assays in the target discovery stage. So we have a very efficient platform in that regard. We can start with the rabbit optimization, then PBMC enrichment. Then using the beacon system, we do single cell analysis and screening to see which ones are the desired clones. Then we can proceed with sequencing of the single B cell colonies. And then we proceed with expression of these recombinant antibodies and delivery to you certain amounts of these recombinant antibodies. So very effective systems for producing antibodies as tools for detection during target discovery. We have also a lot of products for science. We have very complete system for Western blotting. We have excellent gels with very high resolution, very cost effective. That can be used for protein separation. We have a device for staining of the gels, automated staining if that's desired. We have also devices for transferring the proteins from the gel to the membrane, our eBlot L1, and we also have automated system once the proteins are transferred to a membrane, you can use this automated system, EasyWest Lite, to process your membrane automatically, basically adding the primary antibody, secondary antibody, the blocking reagents, your detection reagents, and so on. So it's an automated process for detection. We also have excellent automated system for protein and antibody purification. If you are expressing a lot of proteins or antibodies with affinity tags, the system can use magnetic beads and can very efficiently purify up to 12 samples within 1 hour. So you can purify a lot of antibodies or proteins. If you're performing any screening, you can increase your throughput a lot. You can essentially purify 72 to 96 samples a day with one single system. We also have very strong offering for cell-based assays. We have customized CRISPR knockout cell line service. We can knock out single or multiple genes. We have whole gene CRISPR deletion cell line service, so we can delete the whole gene or any fragment that you would like us to delete. We have customized knock-in cell line service to insert any gene into any cell line that you desire. We have customized CRISPR screening on and off target service. We also have our cell power gene constitutive expression cell line services. Basically, you can constitutively express single or multiple proteins. We have also our cell power gene inducible expression, basically TET on-off system. We also have very strong range of off-the-shelf cell line products. We have over 250 cell lines that are actually off-the-shelf and ready for delivery. And our off-the-shelf stable cell line offering consists of several different portfolios. GPCR cell line portfolio, we have over 100 GPCR-related cell lines. We have FC receptor cell lines for characterization of FC receptors for antibodies. We have immune checkpoint cell lines for cancer research. And of course, we have some cell lines for COVID research, ACE2 and spike protein cell lines. All of our cell lines are single clone. We can offer them as multiple species. We guarantee the performance of each cell line, and we have very stringent QC requirements. So what tools do we have for lead generation and optimization? So as we discussed before, we have multiple capabilities. We can support you in your viral vector developments or non-viral vector developments. Adeno-associated viruses are one of the most used viruses for gene therapy. So we have very strong capabilities in that. We can also do similar services for lentivirus. But basically, we can start with sequence. We can perform codon optimization. We have also very strong cDNA clone collection where you can select the clone of interest. Or we can synthesize it. We can do gene synthesis. Then we can move on from there to the vector optimization and construction. We can construct any vector with any traits that you want. We have different versions of different plasmids available for vector construction. We have different parts that are available that you can insert there. We can do all the cloning and subcloning required for that. And we have our AAV ITR guarantee service. We can also optimize the capsid to enable specific tissue tropism, concentration of the virus to specific tissues or type of cells. We can synthesize the trimer oligos. We can perform mutagenesis to mutate various genes. We can offer you mutant libraries if you prefer to do these mutations yourself using our Gene Titan oligopool technology. So we have very strong capabilities from beginning to end to help support your AAV engineering efforts. We're, of course, very well known being very strong in gene synthesis. We are one of the first gene synthesis providers in the US. We have been offering this service since 2008. We have delivered over 3 million genes so far with over 99% success rate. We have very strong capacity. We can synthesize over 200 million base pairs per month. In addition, we have very easy to use tools such as GenSmart Codon Optimizer. It's a very easy to use software. It's available to all our customers. You can use it to do the codon optimization for your genes of interest before the synthesis, and then easily place the order. We can synthesize the gene of interest, of course, as a single fragment or as a gene brick or gene fragment, essentially. And we can synthesize, especially as gene brick, we can synthesize up to 200 bases DNA sequences. We have proprietary technologies for accurate, efficiently one-step assembly of these gene fragments after the synthesis. In addition, we offer our vector ARC and clone ARC storage systems. You can store your vectors or your clones with us, and you can keep reusing them for the next following projects. I mentioned the GenSmart software system that we have. It's very useful system for optimizing your codon, your gene sequence. It is very easy to use, does very comprehensive analysis using over 200 factors that have been screened and validated. It mimics natural gene sequences, maximizes the expression of the functional protein, and is fully customizable. And again, you can also use it with our vector ARC and clone ARC systems to store your vectors and clones if needed. And we have many very happy customers. Here is one recommendation, one reference from Dr. Lu in Harvard University, who used our gene codon optimization system and found very good results with it. Vector construction, we can construct any vector. We have over 200 commonly used plasmids that are available that you can use and customize. We have over 2,000 commonly used functional parts, and we offer quick access to commonly used databases. And we perform automatic sequence annotation, free plasmid mapping, error screening algorithms, and it's easy drag and drop interface. We basically can solve the vector construction complexity by making it very simple and easy with ordering from GenScript. We can also offer plasmid preparation. We offer plasmid preparation in various quality grades, in various quantities, starting from research grade, industrial grade, and preclinical grade. We can also offer clinical GMP grade plasmid preparation. We have different parameters that we can see in each step, so those are also customizable based on your needs. Our research grade is very economical and very fast. The industrial grade offers higher QC quality requirements, and our preclinical grade offers even higher performance and higher QC requirements. And they're customizable. There are certain add-on items you can add to your QC if you like to. In addition to our plasmid synthesis, we also offer plasmid purification instrumentation. We recently launched our AMAC4 plasmid purification system. This system is very high throughput. It can purify up to 24 samples. It has 4 modules that can be used for the purification and 1 controller. It has very high pipetting accuracy. It has flexible cell culture volume. It can purify somewhere from a few hundred micrograms to multiple milligrams of plasmid per each sample. The run takes only 2 hours, and we deliver essentially transfection-grade plasmid with very low endotoxin levels, a very high supercoat ratio. It's very easy to use because it uses pre-made consumables that contain all the reagents needed for the purification. You can only need to add your bacterial sample and some ethanol, and then push a button, and 2 hours later you have your samples purified. It can purify anywhere from 6 to 24 samples, depending on how many modules you have purchased with the system. If you have low throughput needs initially, you can purchase 1 module that will give you the capability to purify 6 samples at a time. And then with addition of every module, up to 4 modules, you can purify up to 24 samples. So that's a very good, easy solution if you prefer to purify your own plasmids in-house. We also offer our AAV ITR guarantee service. We have optimized ITRs. We also do very accurate sequencing of these ITRs to confirm the sequence. We also have our own strain that we have optimized with ITR regions, and we have compared it with several other commercial vendors of such strains, and our performs very favorably compared to these competitors. As I mentioned, we have the special technology of Gene Titan. We can create microchip-based oligosynthesis, and we can create various oligopools that are useful for various libraries. We can create sgRNA libraries, as I mentioned. We can also create mutant libraries. So using these two, we can very quickly synthesize very large number of fragments and clone them into plasmids or viruses and deliver precision mutant libraries to you for your needs. This is just one paper where we supported customer research to develop various versions of adeno-associated virus to modify the capsid, engineer it specifically for their concentration in the liver, which was very successful, and GenScript supported this with various sgRNA synthesis. We can deliver all these services and products from REO grade to GMP grade, starting from discovery and preclinical research. In the discovery side, we have our sgRNA and HDR templates research grade. We have the easy edit sgRNA, safe edit sgRNA, gene exact sgRNA, gene exact single-stranded HDR template, GENE-1 double-stranded DNA template, and GenCircle double-stranded template. For the sgRNA, the different services vary. The easy edit sgRNA is very useful for very quick and easy experiments, is very cost effective. The safe edit sgRNA is higher purity HPLC purified sgRNAs that are usually used very good for harder to edit cells, such as primary cells, for example. The different templates that we offer, they can serve different purpose depending on your need, and we can work with you to customize the type of template that is being used for your gene editing. For example, the gene exact single-stranded DNA is excellent for minimal cytotoxicity, and it supports very precise knock-ins and minimal off-target effects. However, it's limited in the size of the fragment can be inserted. It's limited to 5 kilobases of fragment to be inserted. Our GENE-1 double-stranded DNA supports larger size fragments up to 10 kilobases, and it's double-stranded DNA with covalently closed ends to stay more stable and more accurate gene editing. Also, it's more suitable for scale-up. And then GenCircle double-stranded DNA template is very useful for very large fragments of 50 kilobases, up to 50 kilobases. It's also circular DNA vectors are very good for very increased efficiency of the knock-in, and they also offer low cytotoxicity and immunogenicity, and it's very good for basically removing any concerns about antibiotic resistance, because if you're using a plasmid, very often they'll have antibiotic resistance genes built in there, but GenCircle is very small. We have removed all these antibiotic resistant genes, and the template is only 429 base pairs. It's very small. We have also excellent sgRNA and HDR template design tools. We have compared our design tools for some popular vendors on the market, and if you can see here in the very top, GenScript's design tools offer very good results with very high predicted efficiency, very high specificity score, and very low off-target effects. While some of the other vendors suffer from low predicted efficiency or from low specificity scores. And here are the results also for the gene editing efficiency for our sgRNAs and for the competitor sgRNAs. And here on the right side is also showing results with different genes. So these are just some results with our easy edit sgRNA done side by side with other popular vendors on the market, offering similar quality of similar service. As you can see, GenScript is usually with higher purity. We have less in mass spectrometry. We show higher percentage of full-length sgRNAs, and we also show higher percentage of full sequence sgRNAs. For the safe edit sgRNAs, HPLC purified. As you can see here from the peak from the GenScript sgRNA is very, very pure, no extraneous peaks. And also here it shows the performance on primary cells. It shows very high efficiency in the gene editing. Very often, mRNA is being used as a delivery mechanism to do in vivo gene editing because it has more transient effect and does not integrate into the genome. And we offer the full service of producing this mRNA for our customers. We can start with sequence. We can perform the codon optimization. We offer UTR design. We have pre-optimized UTRs, or we can work with you to design yours. We also can perform the gene synthesis, anything up to 20 QO bases in a single fragment, or up to 200 QO bases if we use the gene fragment service. Very fast delivery times of 8 days to 14 days. We can also move to the next step with plasmid preparation. Basically, we can produce the vector, we can produce the plasmid in higher quantities, we can linearize the plasmid for you. If you need any special enzymes to be generated, we can design those enzymes, we can express them, purify them, and have them ready for use. We can produce various types of mRNA with our IVT capabilities and our synthesis capabilities. We can produce regular mRNA or circular RNA. Circular RNA is becoming more and more popular lately for advanced capabilities. And then we can also package them as LNP particles. So we can start from sequence, and we can deliver packaged mRNA into LNP particles for you. These are some options we offer in our mRNA production service. We offer different scale. We offer different cupping capabilities. We have poly tail options, no tail, certain length tail, or 100A tail, or something that is different length of tail. We usually recommend it to be over 80 A's in the tail for stability. We offer the use of modified NTPs. We offer different types of purification, silica or oligo-DT. We offer different QC tests for different grades that we can produce. We have customized QC packages for each quality grade. Of course, those are also customizable to your need. And as I mentioned, circular RNA is becoming more and more popular because more stable and very often leads to better expression in the body. So we have different scale for that service. We also offer different IRS option. We have a default CVB3 IRS option, or we can work with you to design your own IRS options. We do different types of purification. We have HPLC purification for a lot of these materials, and also QC tests, variety of QC tests that can also be customized. Poly-A tail is, of course, very important for the stability of the mRNA. So we offer different types. We can synthesize any poly-A tail. Normally, over 80 A's poly-A tails are synthesized, and we can clone the poly-A tail into the plasmid, and we can essentially guarantee the expression of the poly-A tail with the desired length up to 120 nucleotides. These are just some examples of the QC that we do for the different grades of mRNA for our mRNA production. We look at cupping efficiency with mass spectrometry. We look at double-stranded RNA residues with our SWAT blot assay to quantify any kind of double-stranded RNAs. And we look at protein residue assays using nano-orange. These are just some comparisons with different vendors on the market using EGFP mRNA as an example. And you can see that here, if you look at mRNA expression, the GenScript EGFP mRNA compares very favorably. If you will look at integrity, also the GenScript mRNA shows very high integrity. These are some of the customer feedbacks that we have received for our mRNA services. Here, this team at Georgia State University has done EGFP expression in cells with excellent results. This team at Upstate Medical University has done mCherry mRNA IVT expression. Other teams here have done expression in mice. You can see here, and basically in Northwestern University, they have used our mRNA in vivo in mouse models. And then we have very good reference also from researchers from Merck, from XAP, and other companies. So the next I'll discuss our tools for preclinical and clinical development. So we have preclinical grade plasmid preparations with increased QC requirements. We offer animal-free production, animal-free cell culture media. We guarantee the quality and the quantity of the plasmid. We have very extensive QC. We also have a lot of add-ons for the QC. If the plasmid is going to be used in vivo, we can also do mycoplasma. We can do endotoxin testing and removal, and so on. We offer different grades of gene synthesis service. That some of our grades of gene synthesis service can support also preclinical work in filing of IND applications. We QC a lot of parameters here, looking at basically the different qualities of the production, calibrations and verifications. We do online batch management. We have printable electronic forms. And we also can offer audits if we need to be audited, we can support that also. And again, for our preclinical and clinical studies, we have GMP-like sgRNA qualities. We have GMP-like gene-exact single-stranded DNA templates, GMP-like Gene1 double-stranded DNA templates. We also have GMP grades of sgRNA, ESP-Cas9 enzymes. We have GMP grade gene-exact single-stranded DNA templates and GMP Gene1 double-stranded DNA templates. These are some of the acceptance criteria for our sgRNAs. Basically, it has to be colorless, clear. It has to meet our concentration requirements. It has to be very pure with over 80% in the main peak. It has to be very consistent in the mass with less than 0.05%. It has to be sequence correct with 98% or better. Very low endotoxin level of less than 2 EU per milligram and no bacterial growth, essentially sterile. This is just example. We can perform very successfully NGS QC of our sgRNA and detect even single point mutation. Here was sgRNAs were synthesized, one of them with a single point mutation, and was successfully detected. We also do mass spectrometry analysis for the length, and you can see there are no truncation fragments in this sgRNA production. And also we look at the trace for the mRNA trace. In cGMP, we can also produce the HDR templates, single-stranded DNA, double-stranded DNA templates, again with very high quality requirements, no any kind of insolubles, very high concentration, and with very matching our concentration requirements. With specific purity report, over 90% signal intensity has to be in the major peak, over 100% sequence accuracy. very low any kind of residual protein, very low endotoxin levels, no mycoplasma, no detectable mycoplasma, no absolutely sterile, no any kind of growth, pH testing, plus minus 1 unit of pH 7, and very low conductivity. These are just some pictures of our GMP facilities. We have GMP facilities in Nanjing with 21,000 square feet area. We have 5 manufacturing lines in that GMP facility. We also recently built a much bigger GMP facility in Zhenjiang, 400,000 square feet with 12 manufacturing lines. And again, we have GMP dedicated manufacturing facilities. Every production line is separated. We can do flexible batch scales. We have high quality management systems, QA/QC documentation, and regulatory support. We have performed a number of GMP manufacturing batches already. Since 2020, we have performed over 103 manufacturing batches. We have passed over 22 successful audits. We have supported 30 IND projects, and we have had over 6 IND approvals so far. This is just one example of a case study with a company we supported for their human trials with our sgRNA bioarray laboratories. They were performing CRISPR-enabled gene therapy in humans with PD-1 CD19 CAR-T, and that was very successful, and they used our sgRNA for their human trials. This is another example with Professor Zhang performing human trials using the GenScript GMP-grade sgRNA synthesis for GMP-grade for human trials. So we have also supported a lot of plasmid DNA GMP production services. We have done this for over 18 years. We have supported over 7 IND filing with success. We have done over 20 plasmid CMC projects. We have produced over 30 plasmid batches, and we have worked with over 15 customers to produce mRNA for them. And these are just 2 examples of GenScript ProBio CDMO division supporting STEM RNA with their clinical trials, and also Abugen with their clinical trials, producing GMP-grade plasmid batches. And these are some of our services for viral vector gene therapy. We provide services for plasmid production, various services for GMP-grade plasmid production, various services for lentiviral GMP production, and also a lot of services for AAV GMP production of AAV virus. And again, as a company, we can support research for viral vector gene therapies from research and development to process development, clinical, and commercialization. This is just a short overview of all our services that we offer and products. A summary, we just highlighting some of the key services and products because we have too many to show on single slide. But you can see for antibody and protein expression production, we can support everything from early research stage going to preclinical, and then IND filings in clinical, the clinical trials and clinical manufacturing. CRISPR, sgRNA, and HDR templates, also from research grade to preclinical and clinical. Plasmid preparation from research grade to preclinical and clinical. For IVT mRNA, we're still developing our GMP manufacturing capabilities, but we can support research grade and preclinical grade. AAV vector lentivirus peptide synthesis, we can support the developments from preclinical, from research to preclinical and clinical grade and GMP manufacturing. And here below, we have some instrumentation systems. We have excellent instrumentation system for cell isolation, for T cells, NK cells, stem cell isolation for cell therapies. We offer this system as research grade and GMP grade. Also, the consumables can support research and GMP production. We have excellent T-cell activation reagent that we offer in research grade and GMP grade. And we have, as I mentioned, some automated purification systems, plasmid purification system and protein purification system that's usually excellent for research use and scaling up your research in later stages. So why using GenScript? We're one of the biggest providers for gene synthesis. We are very well known for that. We have over 20 years of experience. We have over 235,000 customers in many countries in the world. We have synthesized over 3 million synthetic genes have been delivered with over 95% success rate. We also have a wide range of other services: protein production, antibody production, plasmid production, viral vector production, HgRNA and HDR templates, Cas enzymes in various grades, mRNA production in various grades. So we can support you from the beginning to the end of your development process. Here are some examples of customer success stories, customers publishing and citing the GenScript services or products. In the last few years, we had over 8,000 publications, many of them in very reputable journals. Our customers had very good publications in many reputable journals and citing using the GenScript services or products for that. And thank you for your attention. And if you have any questions, I'm happy to answer your questions.
Thank you, Rumen, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located at the bottom of your screen. Let's go ahead and get started. Our first question asks, can your Gene Titan technology be used to generate NGS libraries?
Yes, we can create NGS libraries. We can create hybridization-based and amplicon-based next generation enrichment libraries, and we can also produce them as ISO 13485 if required.
Great, thank you. Our next question that we have here says, what is the timeline for your cell line development services?
So depending on the cell line service that you choose, our cell line pools can normally be developed within 8 to 10 weeks. Our single clone monoclonal cell lines, they usually take somewhere between 14 to 18 weeks.
Great, thank you. Our next question here asks, what is the difference in delivery time if I order AAV plasmids versus virus?
So plasmid can be delivered very quickly. We can deliver plasmid within 2 to 3 days. All the plasmid that you need for your virus packaging, if you prefer to do it yourself, or we can do the virus packaging for you. That would normally take somewhere between 2 to 4 weeks. Depending on the type of virus and depending on the titer of your virus, we may, and the quantity that you're ordering would be somewhere between 2 to 4 weeks.
Excellent, thank you. Our next question asks, for mRNA, can you help me optimize the sequence for expressibility and stability?
Yes, so we have different capabilities for optimization. We can support the optimization of we can perform codon optimization, we can perform ITR region optimization, we can integrate pseudo entities, we can do 5' modifications with cupping. We can also work with you on further optimization, for example, LNP optimization and stuff like that.
Great, thank you. And it looks like we have time for just one more question. That question asks, can you customize the LNP to target specific cell types or organs?
So we have some LNPs that some formulations we have already pre-tested that can support targeting specific type of cells or specific type of tissues. We can also work with you on making custom LNPs that may better fit your needs.
Thank you again, Rumen Bugaev, for your time today and your important research. Be sure to also check out GenScript's booth located in the exhibit hall for more great information. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until April 3rd, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you for your